Hi all. Today in this lecture, let us discuss about enzymes. In this lecture, we will be seeing the definition of enzyme, characteristics of enzymes, and enzyme classification. Okay, what is an enzyme? Enzymes are biocatalyst. Let us know what is a catalyst. Catalyst is something which increases the rate of reaction without being consumed or undergoing permanent change at the end of the reaction. So let us first now understand this catalyst. So it increases the rate of reaction, it is not consumed in the reaction, and it does not undergo any permanent change at the end of the reaction. Now let us take a daily life example, like take a matchmaker. What he does, he bring the bride and groom together, right? Yeah. And he's not part of the marriage. He just bring the bride and groom together. He's not part of any marriage. Yeah. And after that, after bringing the bride and groom together, he keep many other couples also together, right? Yeah. So this matchmaker is like our catalyst. It is needed for a reaction to occur. Just like how matchmaker is needed for bride and groom to be together, to bring them together, the catalyst is required for a reaction to occur. And the matchmaker is not part of the marriage. Samely, the catalyst is not involved in the reaction. It is not part of the reaction. It is needed for the reaction to occur, but it is not part of the reaction. And after this marriage, he brings other couples also together. Okay, he's not having any change after this, right? In the same way, the catalyst is also not undergoing any change by the end of the reaction. Yeah, this is what catalyst is. So now our enzymes are biocatalysts, which means that are produced by our living cells that catalyzes the biological reactions. In our body, for a reaction to occur, we need enzymes. So our living cells produce these enzymes for these reactions to occur. So these enzymes are specialized proteins. So this is what enzyme is. Now let us see what this enzyme does. It converts the substrate into product. Okay, and this is highly specific. For example, now let us take an enzyme like hexokinase. This hexokinase is specific to hexoses only. It will not act on pentoses. So it is specific. These enzymes accelerate the specific chemical reaction without being consumed in the process. As we already discussed, it is not consumed in the process. The catalyst is not consumed in the process, and it accelerates the reaction. See, now this is substrate. Substrate is something on which enzyme acts. Now enzyme is acting on this substrate. It forms substrate enzyme substrate complex, and after that the enzyme leaves, we get the product. So it accelerates the specific reaction for the substrate to convert into product. This reaction, this enzyme is accelerating. It is not consumed in the process. See, enzyme entered, enzyme exited. It is not consumed in the process. This is what we learned. It increases the rate of reaction without being consumed. See, entry, exit. It does not undergo any permanent change by the end of the reaction. It is converting the substrate into product and is highly specific. This is what enzyme is. Now let us see the characteristics of enzymes. Okay. Almost all enzymes are proteins. I told almost. Okay, what is the exception? We have ribozymes, which are RNA molecules. Even that is enzyme, but that is not a protein. Okay, that is the only exception. Almost all enzymes are proteins. So what they do? They follow the physical and chemical reactions of proteins. Okay, now let us see. Protein means what we generally get in our mind, not just we, what anybody get in their brain is egg, right? So now let us take egg as another example and see all these characteristics. Okay, they are heat labile. I told let us take egg as an example, right? Egg is heat labile. See, our raw egg is translucent and clear. On heating, it becomes white, opaque and solid, right? So, same. Even enzymes are heat labile. By applying some heat, they get destroyed or they get altered. See how egg got altered? Even enzymes get altered by this heat, and they're water soluble. See, our raw egg is easily soluble in water, right? Yeah, that is how we can make all the food item. So the same, even enzymes are water soluble. So these are all the physical and chemical properties of the protein. Okay. Now these enzymes can be precipitated by protein precipitating reagents. I told you enzymes are proteins except that ribosome. So if we add some protein precipitating agents, it will get precipitated. So what are the protein precipitating agents? That is ammonium sulfate or trichloroacetic acid. And this enzyme contains 16% weight as nitrogen. See, now basically the proteins are being the amino acids are building blocks of proteins, right? And this amino acid have amine group in them. So the amine group contains nitrogen. So overall, if we see, the it has about 16% weight as nitrogen because they are made up of amino acids. Amino acids are having amines. Amine has nitrogen. So in overall enzymes, if we see, they contain 16% weight as nitrogen. So these are the characteristics of enzymes. Almost all enzymes are proteins, exception is ribosome. Enzymes follow physical, chemical properties of proteins. That is what we saw: heat labile, water soluble, precipitated by protein precipitating reagent like ammonium sulfate or trichloroacetic acid. They contain 16% weight as nitrogen. This is the characteristic. Now let us see the classification of enzymes. Okay. So enzymes are classified based upon the IUBMB system of classification, which is International Union of Biochemistry and Molecular Biology. So as per this system, the enzyme name starts with EC, which is enzyme class, and followed by 4 digits. The first digit is about class, second is subclass, third is subclass or group, and the fourth is number of that enzyme in this group. The particular number of the number of that particular enzyme in this group. Okay. So by this IUBMB system of nomenclature, there are 7 major classes of enzymes. Those are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases, and translocases. This is recently added. So these are the 7 classes of enzymes. Let us see about them one by one. Firstly, oxidoreductases. So these oxidoreductases, these group of enzymes catalyze oxidation of one substrate with simultaneous reduction of other substrate. So now let us see briefly what is oxidation and what is reduction. See, gain of an oxygen molecule is oxidation. Losing an oxygen molecule is reduction. In hydrogen, that is opposite, like gaining of hydrogen is reduction, losing of hydrogen is oxidation. Okay. Now this AH2, this substrate is losing hydrogen, so this is getting oxidized, and this B is gaining hydrogen, so this B is getting reduced. So oxidation of one substrate and simultaneous reduction of other substrate. So this is what we see. This is what alcohol dehydrogenase do. Alcohol is getting oxidized into aldehyde, and this NAD plus is reduced to NADH plus H plus. Okay. This is what oxidoreductases are. Now let us see transferases. See, in oxidoreductases, hydrogen is getting transferred from one substrate to other substrate. So, any other group other than hydrogen, if it got transferred from one substrate to other substrate, then those enzymes are called transferases. If that enzyme transfer any other group other than hydrogen from one substrate to other substrate, so then it is called transferases. Let us take hexose kinase. This hexose kinase phosphorylases the hexose sugar. So phosphorylation means adding phosphate group. So what it does, it takes phosphate group from ATP and gives to hexose. So what makes hexose converted into hexose 6-phosphate? And ATP get converted into ADP plus inorganic phosphate. Okay. So this is what transferases is. Any other group other than hydrogen is getting transferred from one substrate to other substrate. Okay, this is transferases. Now third class is hydrolases. So what happened in hydrolases is, for a molecule, for a substrate, water is added and then the bond in that substrate getting broken. So water is added and the bond is broken. So now let us take our example. Acetylcholine, we add water. So I forgot to mention the enzyme here, that is acetylcholine esterase. This acetylcholine esterase breaks the bond and it converts acetylcholine into choline plus acetate. This is what occur in hydrolysis reactions. Now this acetylcholine, because of the water action, enzyme action, it converts into choline and acetate. So this particular thing, this acetylcholine, let us just know about this acetylcholine esterase. How is it helping? So generally we need acetylcholine for stimulation of muscle and nerve, right? So what this acetylcholine esterase does is, it stops continuous stimulation of muscle and nerve. So if acetylcholine is there, it will keep on stimulating the muscle and nerve. So this acetylcholine stops that continuous stimulation of muscle and nerve, and it allows the synapse to resting state. So it will break acetylcholine and will give that allow that synapse to come to the resting state. Okay. So this is why this enzyme is important, acetylcholine esterase. If this enzyme is not there, there will be, what happens, acetylcholine will keep on acting. So then we get muscle spasms which lead to paralysis. So this is why acetylcholine esterase is important. This is not needed here while we're talking about classification, but in further you'll get to know all this. Okay. And all our digestive enzymes are hydrolases. Now take anything, like now take protease, it breaks down the protein, and take salivary amylase, it breaks down our food. So all these all these digestive enzymes are hydrolases. Now let us talk about lyases. See, any other mechanism other than hydrolysis. So what is happening in hydrolysis? What are hydrolysis doing? Water is getting added and then bond is breaking, right? And other than water, other than hydrolysis, other than adding water, any other mechanism, if the bond is broken, so there bond is broken, here also bond is broken, there we added water, here anything other than water, if the bond is broken, that is called lyasis. So we will see this particular thing in our glycolysis, which is very important. What happened there? Fructose 1,6 bisphosphate will convert it into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Here fructose 1,6 bisphosphate is a 6-carbon compound. What happens? It breaks into 3-carbon and 3-carbon compound, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay. Here the enzyme is aldolase. Okay, this aldolase breaks fructose 1,6 bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So this is lyasis. Any mechanism, any enzyme other enzyme which is breaking the bond by any mechanism other than hydrolysis. Here water is not getting added, but the bond is getting broken. So this is what lyases are. Now let us talk about isomerases. These isomerases enzymes, these produce isomers like optical, geometrical, positional isomers. Okay. So examples of isomerases are racemases and epimerases. Now here we saw, right, fructose 1,6 bisphosphate is converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate. These both are isomers, right? This glyceraldehyde 3-phosphate converted into dihydroxyacetone phosphate by this isomerase enzyme, which is triose phosphate isomerase. So these are the isomers. These produce isomers like optical isomer, geometrical isomer, positional isomer. Example is our this aldehyde 3-phosphate getting converted into dihydroxyacetone phosphate. And ligases. Ligation is to bring two substrate together. So that is what ligation is. Now here in our example, acetyl CoA, and we are bringing acetyl CoA and CO2 together. We are adding CO2 to acetyl CoA. So here we required energy, that is ATP. Okay, this ATP will get hydrolyzed into ADP, and then this both acetyl CoA and CO2 come together as malonyl CoA. Acetyl CoA is 2 carbon, this malonyl CoA as CO2 is added, this is 3 carbon. So this is what ligases do. What happened? Acetyl CoA and CO2 were brought together and we got malonyl CoA. So the enzyme here is acetyl CoA carboxylase. We are adding CO2 right to acetyl CoA. So the enzyme is acetyl CoA carboxylase. So these are the ligases. And now our last class 7, that is translocases. What this is new class of enzyme that is added in 2018. This catalyze the translocation of ions or any other molecules across the membrane. Okay, from one side of membrane to other side. Some of these translocases use energy. They use ATP. Okay, this there will be some specialized translocases for transfer of hydrogen, some inorganic compounds, amino acids, anything. Okay, so we will read one particular translocase, that is carnitine acyl translocase in our beta oxidation of fatty acids. There what happens is, there will have long chain fatty acids which are impermeable to inner mitochondrial membrane. So this translocase help through this carnitine shuttle system. We will discuss about this in detail in beta oxidation of fatty acids. So what this translocases does, this transfer molecules across the membrane from one side of the membrane to other side. Okay, these are translocases. So this is our last class, that is translocases. Let us just brief everything. Oxidoreductase, this enzyme oxidizes one substrate and simultaneously reduces the other substrate. And transferases, transfer of any other group other than hydrogen, then that is transferases, because transfer of hydrogen is oxidoreductases. And hydrolases, the water is added and the bond is broken down. And lyases, in lyases what happened? Bond is broken by any other mechanism other than hydrolysis. Isomerases, these produce isomers like optical, geometrical and positional isomers. Ligases, they bring two substrates together. Translocases, transfer of molecules across the membrane. So this is the classification of enzymes. So thank you.